So I'm Romain Alderweireldt. I'm a lawyer. I'm specialized in competition law applied in the field of telecommunication. But it's not in this quality that I will speak with you today. I'm also a member of the Belgian Marc Van Association, and I'm a member of something called the Genome for Brussels Consortium. And I'm a member of the VACERN, one of the ERN that you heard a bit about today. And I'm a part of the Belgian mirror group of the 1 Million Plus Genome initiative. And also, and mainly, I'm the co-founder of the 101 Genome Foundation. If there is one thing that you need to keep in mind after this speak is this slide. On this slide, at the center, you have the hand of my son, Aurélien, on the day of his birth. And few months, a bit like with Jackson, a few months after his birth, we heard that Aurélien was really, really sick. So my wife and I, we are reasonably healthy. Aurélien was sick because of a de novo mutation. We never heard about rare disease. We never heard about the disease our son has. And so we went to the patient association. They helped us to understand what was happening with our child. But quite fast, we realized that the patient association, when they are doing this awareness job, they are doing it really well. When they do this welcoming job for the new patient, they are really brilliant. But sometimes there is a gap missing between the patient association and the research. And so we decided to fill this gap, and we decided to create the 101 Genome Foundation with the support of the King Baudouin Foundation in Belgium. When it was done, we created our own biobank. And on the top of this biobank, we create what we call a biobank, a bioinformatic biobank, a genomic cloud, where we gather the digital translation of the biological DNA. Because we are lawyers, my wife and I, we realized that the condition in which the consent is collected, it's suboptimal. And it's really an euphemism to say it this way. So we created something that we call the GEMS app for a specific research project in the context of the disease of our son. And we went back to the patient association, closing the loop. And as of today, more than 125 patients join our cohort, our whole genomic cohort, which made us the largest genomic cohort in the field of our disease in the world. So this is the story that I will tell you now. So all began with this diagnostic odyssey at the birth of our son. And a few days after he was born, my wife discovered that something was going wrong. And it appears after several peripety that he indeed had a rare disease called the Marc Van syndrome. The Marc Van syndrome is a disease caused by a defect or misproduction of the fibrillin. Fibrillin is a protein coded by a gene called FBN1. And the consequence of this mutation of the FBN1 gene is that you have a lot of affliction, musculoskeletal affliction and ocular affliction, but one of the main disorder, it's a cardiovascular disorder. And the fact that you have your aorta that can rupture at any time with this disease. So it's really a bomb on your heart. On the top of it, we discovered some months later that our son was harboring a very specific mutation in his FBN1 gene in a specific area of the gene, which is the area that we can find between the exon 24 and the exon 32. And the people known carrying variation, pathogenic mutation in this area have really a poor life expectancy of only 16 months. We learned that when Aurélien was 11 months. So sorry. So it was a difficult period, a difficult time, and I was totally shocked, and my wife also. And being lawyers, we knew nothing about genomics, we knew nothing about genes, about disease, and we were really lost. And we met this person that you can see on the slide here, who is Professor Guillaume Smits. Professor Guillaume Smits is at the head of the Genomics Human Center of Genetics here in Brussels region, and is the first one who told us about patient empowerment. and he helped us to understand what was happening with our son. And he allowed us to have access to scientific paper, to publication, and to go deeper and deeper in our knowledge of the disease. And with his help, we realized that we were on the verge, at this time we were in 2016, at the verge of a technological revolution. You heard about that this morning. It was really well expressed when we explained the differentiation between the panel traditional analysis of the genes, the exome, you know, this coding part of the genome that is analyzed. We spoke about 2% this morning. We will see here that we're speaking about 3%, but you are speaking about the same thing. The exome, the coding part of the genome, and the GRAL, the complete, the whole genome data. And we spent some time learning genomics, and we realized that if Aurélien had the genomic analysis faster, we could have drugs to protect him faster, and we could have intervened faster. So we understood that the genome was really useful in the context of the diagnosis. But we understood that beyond the context of the diagnosis, the genome was really helpful in the context of the research. Why? Because each sequenced genome, each biological DNA translated into digital DNA helps the researcher to map the complete galaxy of the genome, which is, this morning we spoke about the desert, which is still really unknown, which has all these parts that we need to explore. And we understood that with the genomes, we can go further. And at one point, I was reading this publication called the Resilience Project. And the Resilience Project is a fascinating study. It's a group of researchers who had access to around 600,000 genomes. And they decided to reanalyze these 600,000 genomes. And doing so, they identified 13 individuals in a reasonably good shape, health, who were carriers of pathogenic variants known to be at the source of monogenic disease that are lethal in the infancy, meaning that those people should be dead. They should not be adult and have a reasonable health, in good health. And so with that, I went back to Guillaume and said, what's this? What's this story? How come these people are supposed to be sick and are not sick? And Guillaume explained me the role of what we call the modifier genes, meaning the genes that are able to counteract even some really highly pathological effect of mutation. There are the epistatic genes and the hypostatic genes that can amplify or protect against the disease. And so what I did, I decided to reproduce the Resilience Project in the context of the disease of my son. And I decided to data crunch two different databases. One database with the known pathogenic variant at the origin of the disease, which is created by the French Marc Van Association, which is called UMB FBN1. And on the other side, I used GNOMAD. For the specialist, I use the V2 version of GNOMAD version. And with my Excel sheet, I tried to find an intersection between the two databases, the database of the sick people and the database of the healthy people. And I discovered an intersection. And I went back to Guillaume with that, and he said, yeah, okay, perhaps it's a coincidence, perhaps blah, blah. But have you found in this intersection people harboring pathogenic variant on the specific exon 24 to 32? You remember the specific exon that my son is affected, the specific variation that you can't miss them in your life. If you have that, you are in a deadly situation. And in my intersection, I had 24 people harboring those same kind of variants that are the origin of my son and are obviously not so sick. So with that, Guillaume told me that we had the kind of hint that there were somewhere in the genomes protective modifiers genes, perhaps one or several genes that are able to counteract even the effect of the really highly pathological mutation. And with that, what we can do, if we can identify those genes, we can try to replicate their effect with drugs. The problem is that when we use a tool like GNOMAD, and the specialist knows it, is that we can't go back to the people who join the GNOMAD database. Meaning that, and it is important for all the genomics project, that if you have only a one-way access genomic database, you will lose an important role of it, which is to be able to come back to the patient and do further analysis. On this point, we realized that we could help, and that we could help to gather, and that the scientists working in the field of Marfan syndrome, they were missing a tool, a place where they could share and study genomes and in order to try to find modifier genes. And so with my wife, we decided to create the 101 Genome Foundation and to create a space where the researcher can share and explore the genome in the quest of modifier genes. We did it with the support of the King Baudouin Foundation. We opened a fund, a specific fund at the King Baudouin Foundation. And this is perhaps one of the most important slides of the presentation. We did everything with the help of the philanthropy. So if at one point you want to help us, please don't hesitate. There is a QR code. I wrote a white paper. And with this white paper, with the discovery that we did, we went all over Europe to all the different experts in the field of Marfan syndrome, and we presented them our project and our idea, and they say, yes, this is what we need to do, this is the direction where we need to go, and they decide to join us. So we create a really high-level expert scientific committee, and you can see here you have Gilles de Backer and Bart Lewis, who are really leading the project. You have also Anne de Pape, and of course Guillaume Smits, and Guillaume Jondot from the APHP, and of course Catherine Boileau from the INSERM, who are really the main actors in our scientific committee. So with them, we draft the GEMS project, the GEMS Modifier Quest project, and where we are trying to identify people who are resilient. Even if they have the same FBN1 pathogenic mutation, we have some people on one part of the scope who are really not so affected and live quite well at a long age, and other people who are really sick, really young. And we try to identify those people and create a spectrum to try to identify where could be the modifier genes. So it's why we decided to create GEMS. In order to conduct the GEMS study, we opened our own biobank with the support of the ethics committee from the ULB University and the aggregation from the AFMPS, the Belgian authority. And we created our own genomic cloud. So we created a genomic cloud within Azure. On this cloud now you have the genomes of 125 people having Marfan syndrome. You have 111 exome coming from another study group and 300 panels. So meaning that we have around 536 NGS data from people with the Marfan syndrome. Creating this genomic infrastructure, we created the first link between Macrogen Europe and Azure, which was a challenge, but we achieved it. And we did this with the support of Geraldine van der Roara, who's a Belgian scientist who at the time was working at the MIT, which at the Broad Institute, which was created by the MIT and Harvard, and now she's working for Global Alliance for Genomics and Health. And with the support of Geraldine, we also had the help of Colby, who helped us to create our cloud. And at the end of this experience, he wrote a book called Genomics in the Azure Cloud. So, when Aurélien was really young, we were at one of the many medical meetings that we have with him. And during the conversation with the cardiologist would say, you know, there is a study in Texas, they are studying children like your son. It would be really nice to enroll in the study, here is the paper, take the form, take the consent form, blah, blah. So we went back with the trolley in the car, we put it in the car, we were driving and asked Ludivine, do you have the consent? No, and you? No, I don't. So we lost the consent as soon as we signed it, and we found it really ineffective. When we launched the GEMS study, we realized how difficult is it to create a consent, and that the consent reach the patient. And so with the GEMS study, we decided to do something that we would like to have when they ask us to enroll Aurélien in our study. And so we create what we call this GEMS app that is live. You can use it, like the 125 other people did it already. And you will see it's available in different language. You can do it for yourself or for your children. There's a specific and really educational and pedagogical process that helps you to really understand what you are consenting for and what are the consequences of your consent. And there is also, which is at the heart of the system, a kind of verification, objectivation solution of your understanding of the study you will join. And when you sign directly with the principal investigator, the main investigator. When it's done, there is a specific flow that you receive by post, a kit, a saliva extraction kit, and the participant, they use the kit and they send it back free of charge to the Ghent University here in Belgium, where the biological DNA is isolated. And one part goes in our biobank, and the other part goes for the sequencing, the translation into digital, and it's finally hosted in our genomic cloud. When it's done, the participant on his side at what we called MIDA. And MIDA, it's the portal for the participant where at any time he can find back his consent, which we were unable to do with Aurélien. At any time, he can withdraw his consent. When it's done, the link between his identity and the data is lost. On this MIDA solution, he can have a view of why, in which context, for which kind of study his data has been used. And he can also exercise all the rights that he received from the GDPR, and he can share additional information if he wants, and he can accept to join other study if he wants. This is what we called, and it's not we, it's in the framework of the Australian Genomic Project, they create this kind of solution. I will say not as, I believe that our solution is even better than what they created, but they created control, and they call that a dynamic consent solution. So they went with this idea of dynamic consent, and this is what we propose. A few months ago, the World Health Organization came with a proposal regarding the partage and the sharing of the genomic data, and the World Health Organization emphasized the need to use this kind of dynamic consent model. Our model has been approved by the ethics committee from the UZA, the Antwerp University here in Belgium, the UZ Ghent, and by Groningen. And we received this really positive comment of the Groningen ethics committee saying that they wish that most of, or even all studies use this kind of solution. So as I told you, 125 participants coming from Belgium, France, Luxembourg, the Netherlands, Slovakia, Sweden, Austria, Spain, UK, Denmark, actually there's also Germany, and USA. So we gathered the attention from the Marc Van US Foundation, and they asked us now to go global, that we wider our initiative at the global level. But to get it, we did it only with philanthropy money, and we had a specific target of 100 genomes, and we are already above this target. So now we are really at a crossroad. We decide, what do? We stop or we go global? We are in the thinking process about what will be the next step. So you heard this morning about monogenics and oligo-polygenic disease. So you have this, and we have it with Marfan. Marfan is supposed to be a really highly penetrant monogenic disease where one variant on a specific gene can really affect the health of a participant, of a patient. And you have other disease when you can see that it's only the combination of different variants in different genes that are the source of the disease. Here, you understood that we need to, one, be sure that when we find a variant on FBN1 gene, this variant is clearly monogenic. I will not go too fast, but too far in this, but I believe that you are familiar with the concept of VUS and the difficulty to be sure that sometimes you have in front of a variant that has an unknown signification, and you have to try to categorize it. So with the help of the Professor Guillaume Smith, we use machine learning to create a specific algorithm to help to classify the variant, and this is something that is live also. It's not something that we will do, it's something that we have done. You can go there on this website and you can use it. You can test every potential imaginable position variant on the FBN1 gene and try to simulate if the mutation will be pathogenic or not. This is such a good idea, we believe that it was such a good idea, that even Google had the same. They did it on another scale, but they did what's called alpha missense, and they did it not only for one gene, but for the complete genome, where we only focus on FBN1, but we quite like to see that we had the same idea at the same time. And we insert already the result of alpha missense in our own solution. So you get it, we have the anchorage with the one genome, the FBN1 gene, and on the other side, we need to deploy and to discover the whole network of other genes involved in the disease to try to identify the modifier. And that we did it with the team of Professor Tom Lennart, and we use Orval and Varkov, different machine learning and AI tools that he created with his teams, and we implemented directly in our genomic cloud, meaning that we can launch on our genomic data, the different tools that we created, try to understand and try to find the interaction between the genes. When we came with this idea in 2016, there were no publications about modifier in the context of Marfan. Since the time, there were more and more publications. And now each time we see a publication coming, we can check in our own database if we can see the kind of modifier, potential modifier that we can find there. So, in conclusion, what you need to have in mind is what we try to do is that in the field of disease of my son, there's a drug that works really well on mice, but it's not working at all on human beings. So we need to find another strategy. Doesn't mean that the in vivo mice fishes model, it's from the past. We need to find an additional strategy. We know, and you know, that we use more and more what we call iPSCs, reprogrammed cells, and so we have the in vitro approach. And what we propose to add into this in vitro and in vivo approach, we want to add what we call the in silico approach with our model. And our model lay on 4 reusable pillars. One finance pillar with the Fund 101 Genome at the King Baudouin Foundation, with a legal and ethic pillar with the GEMS app and the MIDA portal that you saw, with a biology, operational, laboratory, and biobank pillar, and a digital and genomic cloud and bioinformatics solution tools already implemented. So we reach our target. We have a model that is exponential, and so we are ready to open it to other rare diseases. Thank you for your attention.
So thank you very much. I have 28 slides for 15 minutes, and I heard already that we're out of time, so I will go quickly. So what I had in mind, having been part of the discussions, there was quite some interest in seeing a little bit what is going on, how we're going to move forward, how we're going to coordinate pilots. And actually, we have a number of projects running that is already doing a little bit of that. So I will explain you one that is linked to the big initiative on cancer, the Europe's Beating Cancer Plan. And the other one is on the mission on cancer. So the plan is more the political side, and the mission is more the research side. And we have 2 projects that are closely linked to that. One is called Can Heal, and the other one is called OncoNGS. I should say OncoNGS started in 20. It's before even the mission, but it's fitting in very well. So, and we talked a bit about procurement. So I will share a little bit with you our journey on the procurement side. Now, the Can Heal project is a project that was aiming at addressing both population and medical use of genomics. There were 2 arms, and when we started to build the consortium, it's quite a large consortium, we rapidly came to the conclusion that we should not apply for the 2 arms separately, but try to bring them together. So we decided to have an answer to, let's say, bridging between the silos of prevention, diagnosis, and treatment, and instead of having 2 applications, only 1. That is also less administration, has also a slight advantage that your budget is larger. Now, it's a large consortium, and I should say some people think that it's a joint action because it looks a little bit of a joint action, but it's a research project. We are 17 countries involved. What is also interesting, and I do not know, is this, yeah? We have more than 45 partners, but it's a very diverse group of institutions that participate. And for some of you that are now already in the discussions in other projects that are running, this is what we would call already a comprehensive cancer infrastructure, but then an international one. The project, we kept the structure that was originally in the call. We built two arms. One is more on public health genomics. The other is more on diagnosis and treatment. And there also we decided, we had this magnificent set of organizations that participate. We asked them, what do you have that could fit within this call? What is running? What use cases could you present? And I must say we received, I think, more than 30, 40 examples. Everybody sharing what they were doing. And at a certain moment, because Alice is here, she said to me, Mark, how are we ever going to make a project out of this? This is so difficult. And then we started to look, and in fact, if you start putting the pieces together, we came to a very nice arm on public health genomics, already thinking about biobanking. We knew about the Genome of Europe. We had the PRS, the polygenic risk scores, and we needed to also try to build on clinical utility, because that's one of the things that everybody's asking if you want to go into implementation. The other arm was more clear for us even. Prevention and early detection was in there. Diagnosis and treat the molecular tumor boards. We were in plenty of projects where molecular tumor boards were being demanded to really get some streamlining, and then treatment follow-up, and then also decision support. It was the area of liquid biopsy, so we decided to go on liquid biopsy, and we wanted to really also build on ethic and legal, and not forget training and education of the professionals, but also of the public. We wanted to involve the patients in surrounding it, and also build on the complete let's say, evidence building towards the next steps, because the project is a 2-year project. You cannot really run large initiatives, but it was giving some insights on what is ready to go into the next step of implementation. And for doing that, we decided to also have a model, and the model was co-created with all the partners. So the elements that we took is evidence, the TRL levels. We have to have some cost effectiveness and some equity assessment, together with quality and external quality assurance, the ethics, legal, and the training. And that we asked all of the use case, use our model, apply it to see where you stand. It's a very strict methodology that we apply. Every 6 months we had a meeting and everybody had to show progress. We had a meeting in Paris 2 weeks ago, and this was really also the first time that we saw where people were. I will skip that, but you have the information. It's very broad. We have things on germlines, on prenatal, on biobanks, on PRSs. We have clinical trial setting up, facing all the problems. We had to set up an inter-laboratory international trial. We have things on molecular tumor boards, and we have also some studies that are really providing us the data for implementing clinical trials on, in personalized medicine. Now, this is just to demonstrate. You set up such an assessment, which is not easy, but all the partners are managing to collect within this phase already information on their use case. It's not complete yet, but at least everybody went to the effort to do it. And if you look at the first assessment where we stand, you see that there is a difference. Some are more advanced at one stage, less advanced in another stage. We're only 1 year busy. We hope by the end of the year to have a complete picture and give a clear-cut, let's say, proposition and recommendation of what could be brought in the next step, either to joint actions, either through the partnership, either through other ways for the next phases. This is what you can expect. So there will be guidelines, there will be tools, there will be practices. We have some ethical and legal new elements that we would add. And what is very interesting, we have modules on training and education that are e-driven and that are really an additional tool that obviously we do not see in many instances, and it starts to be in different languages, because that's very important also that the training modules are in the different languages of the countries. Now, one thing that came up of the Paris meeting is the question, shouldn't there be an independent agency that decides a little bit what is the validity of the novel technologies? It was just the first idea. We would like to extend the training scope. Everybody wants us to include patients in the projects, but that is sometimes often tedious. But maybe we should also look once at the relatives, because they can also give quite important information. And then we should definitely focus on youngsters to get them more, the schools involved in our thinking on how to communicate on all these efforts. The use cases that we identified are listed there. And then gradually, there was this oncology decision support tool, and then we were thinking, we were in Paris, you all are aware about Watson for oncology, maybe we should start thinking on a Pierrot for oncology. But okay, we had already discussions, that's easily said, but it's a quite challenging endeavor. And then of course, we said that we had to align and integrate. And then I come to the next slide. This is an overview of the projects that Cienzano is in. So we are now running present in about 17 different projects. And the next one that is upcoming is the Joint Action on Personalized Medicine. And if you look at that, you can see that there are numerous things. Wait, I should go back. No. It's this one that I have to like. We see a lot of pilots. The ones that in orange are pilots. So there is in many projects now pilots ongoing. But what is important, there are a number of structures that are being built. There is a network of comprehensive cancer centers, so hospitals. There's expertise network. There is the tools in the Joint Action on Digital are being tested in other frameworks. There is cancer register are being implied. And then you have all these structures here that are more from the digital side. And all these things are going to be delivered at a certain point in time while we are going to start running our Joint Action Personalized Medicine. So I see the Joint Action Personalized Medicine as a particular opportunity to bring all these things together. So there's a lot of people that are in the different projects already. So there is an opportunity to really use this joint action, which has quite some budget. It has 30 million euro as budget. So we could use this as a tool to start integrating all these projects. I didn't forget here IHI. At a certain moment, if you start really large trials on personalized medicine, you cannot live without the industry. So we are now learning as a consortium how to deal with the IHI. And then you saw in the slide also that we had OncoNGS as one of the, let's say, the projects that should deliver something that may be interesting in the Joint Action Personalized Medicine. And OncoNGS is what we discussed in the panel, is a pre-procurement, pre-commercial procurement initiative that actually, if I remember well, we started it during the IC Permet. It was during IC Permet that we started thinking, yeah, we need some kind of tool that is allowing us to have an integrated and common way of looking at this complex tumor profiling. And then it was the Commission who started to think, yes, that might be interesting, and they opened the PCP initiative, something that I was totally unaware what it was. We said, yes, it's a tender. But we got substantial budget for the project, 12 million. Now, the 12 million is mainly for the development, because the PCP is a partnership between the market and a set of, let's say, institutions that are willing that they perceive an unmet need. And the unmet need that we had provided is that we didn't have a pan-cancer tumor marker analysis that allows efficient, and with efficient I mean also sustainable, so at an affordable price, molecular DNA and RNA profiling. We, at that time, we were thinking about 2020, 2019, 2020. We were saying liquid biopsy would be in 25, 26 be important. It had to be on NGS, and it should include not only the sequencing, but also the analysis, the reporting, and the interpretation. This is the consortium, so we are partners from Belgium, France, Germany, Italy, and Spain, and we are supported. These are mainly hospitals, and we're supported by a number of organizations. First of all, Aquas, who helped us to understand the PCP, but we have also the Cancer Register, we have l'Institut National de Cancer, we have research partners, and we also have patent IP partners. And this is the process. I will not be able to explain you, but there are in theory 3 phases. It's competitive. You start in phase 1, where you ask to design, with 4 partners. In phase 2, there is an analytical performance testing. You have 3 partners. And then in phase 3, where the clinical performance is tested, you remain with 2. This is the schedule. What you have, you define your unmet need, you run your PCP, and at the end, you hope that the solution is there that solves your unmet need. Now, I should say, this is just to where we are. We are in phase 2. And I must say, I cannot really disclose what actually the partners are doing, but a PCP is you procure research and development. So people really have to develop things, and I must say the solutions that are now developed by our 3 partners that are indicated here, Agilent, KU Leuven, and Hufheid, there are really, some of them are really looking disruptive, maybe even too advanced to be in the market, but it's really showing that this kind of approach is bringing innovation closer to patients, because you are directly with people who are in a setting where it's going to be used, and there is also, we imposed a cost, a price to it that is affordable by healthcare systems. So I will skip that. This is just to say what is the market, because that is important if you have a PCP that is a market. We estimated at about 100 to 200,000 tests a year just in the clinic. That is not thinking about when you would go in the population and liquid biopsy being used in population screening, maybe with tests that are derived from OncoInGes. Now this is my final slide. So where are we? We have these building blocks are being produced. They're practically all there already. I didn't cover all of them. We have the projects that are being launched. We have the partnerships, because the projects that I indicated, I would say, are more policy support projects. But there are gaps, and that will be clear from our assessment that there is still research needed. So what is missing? We should be able to look for complementation with the partnerships. And then we will definitely, in the next joint actions, try to bridge between all these things, considering the legislations that is there. And the question that we now have, how are we going to build the engine, the driver? Because I hear in every project, people tell me you have to make synergies, but you have to find an instrument, a way that doesn't consume too much time. So it should be something that is built in the processes of the projects that you're running, instead of asking additional things, because that is probably going to be demanding too much. So I think I can leave it here.